Hello, everybody. Welcome to Immunology. My name is John, and this is my third time doing this class as SI. And this is probably my favorite class to do SI for because I find this topic really interesting, even though it can be a little bit hectic. I am a graduated student. I'm starting med school in August at MSU. So I was pre-med, but I'm graduated now. And I sound a little stuffy because I actually have COVID right now. But I think I'll be all right. So yeah, let's talk about some stuff. Stress management. So a lot of people kind of get stressed out in this class, especially with us being back in person and sort of having more demands about where you need to be and when you need to be there. In general, be sure to reach out to your friends and your family and consult CAPS if you feel so inclined. CAPS is Counseling and Psychological Services, and it's available for free to every student. Confront your stressors head on. Create and follow a plan for problem solving. Focus on goals instead of the stressor. And meditation and exercise. Take care of yourself because the semester can be really overwhelming and stressful, and immunology doesn't exactly help. You guys can all do this, and people have done it before. And if you come to SI, you're especially, set up pretty well to pass this class. But make sure that you're taking care of yourself so you can learn. And in immunology, make sure that you plan ahead and you ask for help early. And most people in this class are going to need help with this class, like one-on-one. That's just an expectation. There's a lot of material, and it's really confusing and sort of hard to organize. You can reach out to me at this email here or Dr. Sanders. We're probably going to expect you to study for 3 to 5 hours per week on top of just learning stuff. That is to reinforce what you know. And attending SI and requesting tutoring, just send me an email right here if you want to request tutoring. If you attend SI, you're in a lot better shape than if you don't attend SI. And a lot of students come to every SI session in this class in particular. Compare that to micro, we don't get a whole lot of attendance. In this class, it's usually a majority of the students in the class. Read your textbook, and I do have a textbook PDF. If you don't have the textbook for yourself, I can email you the PDF if you send me an email. And in this class, it's important not to procrastinate because the material can be very confusing and involved. So make sure that you plan ahead in this class and you stay on top of things. So how do you gain immunology? Your grade is largely going to be just a function of the time that you invest in this class, and that includes SI. Coming to SI is a really efficient way to actually learn this material and have it explained to you, so you don't have to do 100 Google searches to figure out what is happening. You can just have, a good explanation that's down to earth, and that's kind of my goal in SI with immunology. My goal is to never waste your time in this class. If you spend time studying and reading, that helps too. Organization is important in this class, both mentally and physically. So making an effort to organize the material, like making a spreadsheet for this class, and I'll show you a little bit about that. It will pay off big time. This class can be really hard to sort of understand what's what. Logic and reason is a good way to gain immunology. Place them above memorization whenever you can. Ask why. What is the significance or purpose of this? Don't rote memorize in this class whenever you can avoid it. Learn how things work together instead of just memorizing facts. SI and tutoring. Efficiency. Focus on your high-yield content. This is another pro of coming to SI, is I tell you what the high-yield content is directly. I'm going to tell you what you need to know. Don't commit complicated, unnecessary things to memory. You're going to learn a bunch of pathways that don't matter. Only a few things in them may matter, or maybe none of them matter. Focus on the outcomes of processes like signaling and cell behavior modulation, especially if I tell you to focus on the outcomes. Some pathways you'll have to know, but others you won't have to know the full start to end of it. And reading. A few pages per day is good. I know that Janeway is like a really dense textbook. I actually read it when I took the class and it helped me a lot, but I also didn't go to SI. So if you aren't here, you should probably be reading your book. So you can't learn everything about B cells at one time, for example, because B cells interact with a lot of other cells. They interact with T cells and they make, oh, we'll get into that later. But essentially the knowledge is going to build on itself a whole lot. Therefore, if you keep a document or a spreadsheet where you list everything you learn about each cell type, that is a good idea. You should make categories like the cell's functions, their secreted products, like things that they make, surface molecules and markers. This is all a very good idea. This is something that I recommend that you do. Other tabs for your spreadsheet might include proteins or substances that are secreted by cells, lab techniques, processes like, for example, the process of affinity maturation, which we'll talk about, and CD codes. There's a template for immunology on Blackboard. Okay. And it can be hard to consult Google for immunology help. A lot of times you're either going to find something that doesn't explain something at all, or it's way too in-depth. It's hard to find a good explanation of stuff out there, especially at first when you don't know what anything is. Lots of this will show again in other contexts, so we're going to be repeating a lot of the material, but expanding on it and explaining it in a different way over and over again. That should help learn, but the point is, don't freak out early on in this class. It is a lot of material, but you're not really expected to grasp all of it at a super deep level immediately. Okay, so focus on the level that we're presenting it at. And to get started then in the course content, many things can potentially kill you, meaning that lots of response types and a very high complexity is necessary for defense against invasion. And with adaptive immunity, you get immunological memory, and you have a very high antigenic specificity, meaning the immune system is capable of recognizing virtually any molecular structure. Anything that is foreign can be targeted and killed. If the bug was killed off last time, you have memory for that bug, and it is dead even quicker the next time it invades. These are some of the things that complicate the study of immunology despite being beneficial for survival. So some terms and background for immunology. An antigen is any distinct molecule that can be recognized by an antibody or by an adaptive immune cell receptor. And that recognition is going to occur with very high specificity. Antibody-antigen interactions are some of the strongest protein binding activities that are known. The antigen is going to have chemical characteristics that enable the very highly specific binding with little or no cross-reactivity to other antigens. This antibody, this blue antibody here, antibodies are Y-shaped proteins like this. This antibody binds to this antigen. It's not going to bind to any other antigens, just this one. And that is because it is so specific for that antigen. The epitope is the very small specific location on an antigen that a particular antibody binds to. So this epitope right here on this antigen is the location that the antibody actually recognizes. Okay, so it's a single antigen is usually going to contain a large number of epitopes. So multiple antibodies are going to be able to bind to the same antigen at different sites. The paratope is the small specific location on the antibody that binds to the epitope. So your paratope is located right here at the tip of the antibody where it binds to the antigen. And in sort of a different subject, the MHC, or the major histocompatibility complex. You're going to hear this a lot. At first, it was a set of genes that were linked to tissue histocompatibility. And this means that people who had different copies of the MHC genes were unable to transfer organs to, or they were unable to donate organs to recipients who had different MHC genes from them. So at first we knew that the MHC was able to limit the ability of a person to donate their organs to a recipient, but we didn't really know what the MHC did. And the MHC, it turns out, is a set of genes that encode transmembrane proteins by the same name. So the MHC genes encode MHC proteins. And MHC proteins, as we later discovered, are used to present peptides in a process called antigen presentation to T cells. Okay, so these MHC proteins are going to bind to peptides in the cell, and then they're going to take those peptides to the cell surface where T cells can come and swing by and see if that peptide inside the MHC protein is foreign. Okay, MHC proteins, as it turns out, belong to two distinct classes. MHC class 1 proteins, or MHC1, these proteins are expressed on all nucleated cells and platelets. The only cell that they're not expressed on is red blood cells, which do not have a nucleus. And they tend to be loaded with endogenous peptide antigens. These are peptides that are produced inside the cell. So usually MHC class 1 proteins, if they're loaded with a peptide and that peptide is foreign, that means the cell is infected with a virus or something of that nature. The cell is actively infected. On the other hand, MHC class 2 proteins, they are expressed only on specific cells called professional antigen presenting cells, PAPCs, or just APCs. Sometimes they're just called antigen presenting cells. MHC class 2 proteins are loaded with peptides that are exogenous, meaning they were taken up by the cell from the external environment. So the pathogen was not infecting the cell that has the foreign peptide on the surface, the exogenous foreign peptide on the surface, peptide inside. And the antigen cells classically include your dendritic cells, macrophages, B cells, and monocytes. And this is all stuff that this is all very important material. And that's why I want to get it out of the way as soon as possible, because it becomes really confusing for students, like right away. Okay, so we're going to revisit all of this. I know it's already confusing, but that's okay. This slide here is where I kind of start the actual material besides just background terminology. And the reason for that is it shows you how things normally are in the body. So what we have here is a blood vessel that feeds a tissue of some kind, a connective tissue. Excuse me. Like I said, I have COVID, so this blood vessel contains cells, and the blood vessel itself is actually composed of the endothelium, which you see here. These are endothelial cells, and they are surrounded by a basement membrane, and beyond that is smooth muscle, which is composed of smooth muscle cells. Okay? And inside the blood, you have a certain list of cells. You've got these PMNs, these polymorphonuclear leukocytes. You've got lymphocytes, platelets, monocytes, clotting factors, and other serum proteins, eosinophils and basophils. Okay, we're going to visit each of these cells one by one in a minute. But then outside the vessel, you have tissue cells that are just hanging out in connective tissue. And it turns out that the job of the tissue cells is to survey for infection. So they're always looking for infection. Now, that is true for immune cells, but not so much for fibroblasts. Fibroblasts, they're not really immune cells. They just make extracellular matrix. So they secrete collagen and all that stuff. Mast cells are immune cells, however, and mast cells are going to be surveilling the tissue for any signs of infection. Macrophages are also immune cells, and they're going to be doing the same thing. However, the means by which they do these things and how they bring about changes when they detect pathogens is different between these two cell types, and you'll learn about those differences. Okay, And the connective tissue matrix in the connective tissue is composed of elastic fibers, mostly the protein elastin, and then you've got collagen fibers and proteoglycans. That's most of the constituency of the extracellular matrix. Okay, so this is how things normally are. Some things to notice is that at normal steady state conditions, at homeostasis, right, these endothelial cells are connected by tight junctions. Okay, so there's not a whole lot of ability of blood, fluid, or proteins to leak out into the tissue between these cells because it is a tight junction, right? Technically, it's a desmosome, but the point remains. For the most part, during steady state conditions, there is not a whole lot of leakage of blood fluid into the tissue. Okay, it's a very small amount, but it's not very significant. The other thing is this smooth muscle here maintains the vessel at a certain diameter that it normally exists at. Okay? Some of these things are going to change when pathogens are detected. So some more terms then. Cytokine. A cytokine is a signaling protein that is secreted by immune cells and sometimes other cells for the sake of modulating any aspect of immunity whatsoever. Okay? So for the most part, these are made by immune cells and they act on immune cells, but they can also be made by other cells and they can also act on other cells. And they're a very broad family of proteins. But the take-home message is that it is a protein secreted by a cell that acts on another cell to modulate immunity. The scope of the role that they play is very large. You could say that the cytokines are the reason for every bad and good thing. Cytokines bind to receptors that usually heterodimerize or trimerize and signal by the JAK-STAT pathway. That's true for like 9 out of 10 cytokines. They signal by the JAK-STAT pathway to cause changes in the target cell. Cytokines can act in an autocrine, paracrine, endocrine, or juxtacrine fashion. So autocrine means that it acts on the same cell that secreted it. Paracrine means that it acts on a neighboring cell within a close range. Endocrine means that it circulates through the blood and acts on a distant cell. And juxtacrine means that a physical cell-to-cell contact is required for the action of the cytokine. Sometimes cytokines are grouped into 3 broad classes by function. And there are the pro-inflammatory cytokines, the anti-inflammatory cytokines, and the hematopoietic cytokines. That means that they are differentiation and or growth promoting cytokines if they're hematopoietic. More usefully though, and the nomenclature tends to prefer this grouping, more usefully they are grouped into 6 families based on broad homology and protein structure. You have the interleukin family, or IL, the tumor necrosis factor family, TNF, the interferon family, IFN, the chemokine family, which directs cell migration and cause chemotaxis, the transforming growth factor family, or TGF, and the colony stimulating factor family, or CSF. And one other term to know is lymphokine. A lymphokine is a cytokine that is produced by a lymphocyte. A lymphocyte is a specific type of white blood cell or a specific type of leukocyte. A white blood cell is a leukocyte. Okay? Lymphocytes are a specific type of leukocyte. And they include T cells, B cells, innate lymphoid cells, and natural killer cells. Okay? Again, we're going to revisit all of this so many times, so don't freak out just yet. It's really flustering at first, but don't worry, because you'll get it. So, inflammation is one of the first things that I like to talk about because it's something that sort of governs all of the innate, or I should say all of the immune system's behavior. Inflammation specifically occurs when cells of the immune system, i.e. leukocytes, become activated by the signs of microbial presence, host cell death, or disturbance of physiological structure or function. So they may detect infection, they may detect necrosis of host cells, and they may detect tumors or traumatic injury. All of these things can induce inflammation. They can trigger inflammation. This is how it normally works. Sometimes it can go wrong, but this is how it normally works. These things trigger inflammation. So cells of the immune system become activated by these things, and they then produce cytokines, and they produce other mediators like histamine, the eicosanoids, etc. And all of these mediators promote physiological changes to the cells and the tissues that the mediators act on. The symptoms of inflammation are the result of cells receiving these pro-inflammatory mediators that were produced by those activated immune cells. And the 5 symptoms of inflammation, when it's localized acute inflammation, they are redness, swelling or edema, pain, heat, and loss of the functions normally performed by the tissue. Okay, so you see here a typical case of inflammation. You have a swollen leg. It looks really red. I'm sure you can imagine this is painful. And it would feel very hot if you were to touch it. And it's not far-fetched to say that this skin is not going to be producing vitamin D and doing all the things that it can normally do because it is inflamed. Okay, it's too busy taking care of problems to perform its normal functions. Okay, that is acute inflammation. Acute inflammation is a little bit different from this thing called chronic inflammation. Chronic inflammation is distinguished from acute inflammation by the length of time that it takes for it to happen, but it also differs in histological features and in which cells and cell products are involved. Okay, so chronic inflammation takes longer to progress. It's sort of less of a fast onset and more of a long smoldering type of thing. And it also is different in terms of what cells are involved and what they're doing. Okay, we'll talk more about chronic inflammation later, but for now we're going to focus on acute inflammation because that's immunology's core. So inflammation is important but imperfect is the title of this section of the PowerPoint. This is sort of what inflammation looks like, acute inflammation. So bacteria are recognized by macrophages in the tissue. That's where most infections happen. They happen in the tissues. Not in the blood vessels, but in the tissues. That's usually where they start. And they act on macrophages. They're recognized by macrophages. And those macrophages then release cytokines and chemokines. Okay? Cytokines and chemokines. The cytokines are going to act on the vascular endothelial cells. Okay? And that's going to vasodilation and increased vascular permeability. Vasodilation is because the smooth muscles that surround the vessel relax and they enlarge or dilate the blood vessel. And this results in heat and it results in redness because you have an increased blood flow to the area. Increased vascular permeability is a big long term, but what it really means is that these endothelial cells move apart a little bit. And so the vasculature literally becomes more permeable to fluids and such. So you begin to have a leakage of fluids from the blood into the tissue. And with fluids comes blood proteins. And the blood has certain proteins in it that are capable of helping to kill bacteria and are capable of also helping to activate macrophages once they recognize bacteria. And also with the increased vascular permeability and the activation of these endothelial cells, leukocytes from the bloodstream can begin to leave the blood and enter the tissue where they're going to try to fight the infection. Okay, so they're actually going to squeeze out of the blood physically between those endothelial cells. They're going to squeeze out and try to get into the tissue where they will then fight the infection. Okay, and they can only do this when the vasculature is inflamed like this. So inflammatory cells migrate into the tissue and they release inflammatory mediators that cause pain. Some of the pain actually comes from the squeezing out of these cells, but also a lot of the inflammatory mediators themselves act on neurons that sense pain and they sensitize them to pain signals. Okay, so that's where some of the pain symptom comes from. Lastly, blood clotting is a typical feature of inflammation. Usually inflamed tissues are more prone to blood clotting. Okay, so acute inflammation then, it involves vasodilation because the smooth muscle cells relax and that enlarge the blood vessel. And this leads to slowed blood flow and increased blood flow to the inflamed area, to the inflamed tissue. It also involves endothelial cell activation which leads to increased vascular permeability, and the upregulation of adhesion molecules that these immune cells, these leukocytes, can use to leave the bloodstream and enter the tissue. That's what those adhesion molecules are for. Acute inflammation also involves increased fluid leakage into the tissue from the blood, increased effector cell access, meaning white blood cells can leave the blood to enter the tissue, and the recruitment of effector cells by those chemokines that were made by the macrophage and by other mediators. It's not just chemokines, it's also other molecules that signal to these leukocytes where they need to go. Okay, so this is an overview of what inflammation actually involves. It's mostly a vascular thing, but it's a vascular thing so that it can become an immune thing. So here are some of the mediators of inflammation. You have histamine, which is released by mast cells, basophils, and to some extent by platelets. Histamine drives a lot of the early inflammation because histamine released from mast cells is a near instantaneous thing. Histamine is pre-synthesized and it will be released when the mast cell is activated. Okay, so it's actually pre-made. And then you have eicosanoids. Eicosanoids include the prostaglandins, leukotrienes. These only show up after a minute or two because they are organic compounds. They're small little organic compounds that have to be enzymatically activated in order to be released from the cell and cause a response. However, cytokines take at least 1.5 hours before they're released and cause any kind of effect. So cytokines take a lot longer than histamine or the eicosanoids. Okay, so this does begin to take a little bit longer. And we'll talk about some of the cytokines probably in the next slideshow. Inflammation is a protective response. You can see that those leukocytes, they're heading toward infection by leaving the blood, and they're being recruited into the infection where they can then eat up some of those bacteria and shred them to pieces. And also they can provide help to the immune cells that are already present in the tissue. However, although inflammation is intended to be protective, it has a lot of issues. It is nonspecific. Regardless of what kind of infection you have, you're probably going to have the same sort of inflammatory pattern. It can directly or indirectly kill your cells, and sometimes a lot of cells can end up dying. The process of leukocytes leaving the blood and entering the tissue and getting to where they need to go results in the apoptosis and the necroptosis of a lot of the cells that's sort of in the way. If inflammation becomes systemic instead of localized, right? Usually you're looking at a little area where the infection is present that turns red and gets hot and is painful. However, sometimes those pathogens can enter the bloodstream and they trigger a systemic response. They trigger the bloodstream release of some of those mediators. This can cause widespread fluid leakage, meaning you lose a lot of blood volume into your various tissues. And this can result in sepsis and septic shock. Your heart can actually stop from systemic inflammation, which is an issue. Your heart stops, you die. Inflammation can be inappropriately triggered, directed, unregulated, or inhibited by pathogens. The accompanying cell recruitment can lead to the release of an excessive number of inflammatory mediators and a feedback loop. One example of this is in COVID, which is caused by the virus SARS-CoV-2. And in this case, you have an ineffective early response to the virus that doesn't control the replication of the pathogen, but recruits a really large number of immune cells that then release more inflammatory mediators and recruit more immune cells. And it results in a big positive feedback loop that causes a massive amount of inflammation and culminates in respiratory distress. You get a ton of fluid leakage out of the blood and onto the alveolar surface, and that impedes the oxygenation of blood. Inflammation can cause cachexia, or wasting of muscle and fat, the mobilization of basically nutrients from stores. And yeah, if you have chronic inflammation, this is especially bad. You can begin to look rail thin. It can cause pain, it can cause fever. Fever can result in brain damage or seizures, and it can also result in you feeling really uncomfortable. And it can cause a whole host of other problems that we care about in medicine. And inflammation can be associated with the development of autoimmunity because it can accidentally allow self-reactive lymphocytes to become activated. A lack of resolution of inflammation can lead to chronic inflammation, which can result in fibrosis or scarring, cancer, amplification of the response, autoimmunity, etc. Okay, so you can see how many issues inflammation can begin to have. It is a double-edged sword. On the one hand, inflammation is necessary to limit the ability of the pathogen to cause disease and spread and replicate and make you feel sick. But on the other hand, inflammation can actually directly cause a lot of damage, and the immune system can't exactly know whether it's doing too much because it occurs at a cellular and molecular level. Okay? There are two phases of a typical immune response to an infection. Okay, and they have distinct functions. There's the innate immune system, which occurs first, and then there's the adaptive immune system, which occurs second. The innate immune system is extremely evolutionarily ancient. Okay, these have been around for forever, pretty much since the first eukaryotes existed, and probably even in bacteria, we've had innate immune system mechanisms. And the adaptive immune system only evolved relatively recently in evolutionary time, within the last 100 to 200 million years. The innate immune system's job include the prevention of inoculation of bacteria and other pathogens into the body through physical, chemical, and microbial means. So we have a lot of first lines of barriers that prevent bacteria or fungi or parasites or viruses from ever even being able to get into our bodies, right? We have skin, we have tears, and we have saliva, and we have mucus, and we have all these different things that prevent the bacteria from ever reaching you. But it's also a little bit more refined than that. It performs surveillance through pattern recognition receptors, which are broadly specific receptors that recognize pathogen-associated microbial patterns. More on this in a bit. So it performs surveillance of the tissues that should be sterile. If a bacteria is present in a sterile tissue, then that's an issue, right? For example, your muscle should not have any kind of non-host cells in it. And if it does, it's going to trigger an inflammatory response. The innate immune system also limits pathogen replication and spread through the tissue until adaptive immunity can finally kick in. So adaptive immunity has to be mounted before it will finally do anything. It takes at least a few days for adaptive immunity to begin. The innate immune system initiates inflammation, meaning it also recruits leukocytes and lymphocytes to the site of the problem. And way later on, after the cessation of the noxious stimuli, meaning after the infection has been resolved, the innate immune system has to repair the damaged tissue because the immune response and the infection are both going to cause damage to the tissue. There's no way around it for the most part. The innate immune system also activates the adaptive immune system, and that is through antigen presentation, like I told you with MHC2. This is the bridge between the innate and adaptive immune systems. More on this in a bit. And then you have the adaptive immune system, which finally can kick in after the innate immune system tells it to do so. The adaptive immune system, after it kicks into swing, it then activates the innate immune system. You have T cells producing lymphokines that activate your innate immune cells, and you also have B cells producing antibodies that will recognize the pathogens and mark them for destruction and do all these other crazy things. You also have the production of highly specific antibody, which are made by plasma cells. By the way, plasma cells are the final form of B cells. And the adaptive immune system causes direct, highly specific killing of infected cells. You have these cells called CD8 positive cytotoxic T lymphocytes, or CTLs, that go and kill cells that are infected with viruses. And they're very good at recognizing what cells are infected and what cells are not infected. The adaptive immune system is also responsible for immunological memory. you have memory T cells and memory B cells that can rapidly become activated anytime the pathogen is reintroduced after this primary infection. The adaptive immune system has very high specificity to the pathogen. So it's much more high in its specificity than the innate immune system. And it can undergo affinity maturation, meaning it gets better every time. Antibodies made during the immune response actually increase in affinity throughout the immune response, which is a really fascinating topic that we will get into in this class. And the adaptive immune system has two major modules. At least historically in immunology, we have sort of recognized two major modules of defense by the adaptive immune system. There is humoral immunity, which is mostly mediated by antibodies and is most effective against parasites and extracellular pathogens. And then there is cell-mediated immunity, which is mostly mediated by these killer T cells and is most effective against viruses and intracellular bacteria. So if the pathogen is replicating outside of cells, it's going to be taken out by humoral immunity. If the pathogen replicates inside cells, it's going to be killed by cell-mediated immunity. Okay, so this is all just a brief overview before we get into each of the cell types. Again, don't freak out just yet because it'll all click later on once you see the specifics of each of all of this. So, innate immunity is first, and it involves all the things that you see here. It involves barrier protection, inflammation, phagocytosis, and limitation of replication of the pathogen. And then antigen presentation occurs, and that's when the adaptive immune response is recruited and kicks in. Dendritic cells are major antigen-presenting cells that activate T cells. The T cells are sort of the central mediators in the adaptive immune response. You get a proliferation of B cells and T cells around days 2 to 3, maybe 4 or 5. And finally, effector T cells and plasma cells, which make antibodies, form. And that is when you get the final finishing off of the pathogen that hopefully has been inhibited to some extent by the innate immune system. So this whole time, the innate immune system is sort of holding its own, waiting for the adaptive immune system to swing in and play the hero. Okay? The adaptive immune system is required anytime the innate immune system cannot fully eliminate a pathogen without help. Okay? And for the most part, the adaptive immune system is working by activating the innate immune system to be even better at its job. And it also has these killer T cells that can kill virally infected cells and such. Okay, so this is another overview of it. Take a look at this on your own time. So our first cell type is the macrophage. So immune surveillance is carried out by the innate immune system, among other things. Macrophages are one of the most important cells that carry out surveillance. They are tissue-resident phagocytes that are always ready to detect microbial infiltrators. So they live in tissues instead of in the blood. They live in the tissues. Okay, they don't circulate, but they just stay in place, sort of looking for trouble. And they're phagocytes, meaning that they are really good at taking things up and shredding them to pieces once they've taken them up. They're always ready to detect microbial infiltrators because they have a very, very wide range of receptors that can recognize these many different classes of pathogens through conserved ligands, right? For example, all bacteria make proteins that start with formylmethionine. A lot of fungi have a cell wall that contains beta-glucan. And there are other similarities with other pathogens too. Those things can be used by innate immune cells to recognize that there's a microbe present. So how are microbes detected then? By these things called pattern recognition receptors, PRRs. And this is a broad class of innate immune receptors that recognize microbes. Those PRRs recognize ligands that are broadly referred to as pathogen-associated molecular patterns, or PAMPs. These are things that are contained within the molecules made by pathogens that are sort of conserved against, like, across that pathogen class. Like I said, formylmethionine residues present in bacterial proteins. We don't have those, but bacteria do. So those can be recognized. PRRs can also recognize damage-associated molecular patterns. And these are sort of molecules that are released from self cells that have been killed or that are dying and are in distress. And that can also be an indicator that there is an infection or a major problem that's present. And that can activate those PRRs. And it can recognize other things too. The binding of a pattern recognition receptor to a pathogen-associated molecular pattern, in other words, the binding of a PRR to a PAMP, it activates cells in the innate immune system, including macrophages. And for macrophages, this triggers three major things. First of all, it triggers the phagocytosis or uptake of the pathogen, and then the subsequent degradation of that microbe, or shredding to pieces of that microbe inside the cell, by the endosomal lysosomal pathway. And this involves a low pH, so we actually sort of bathe the pathogen in acid, and that can kill it. It also involves hydrolytic enzymes that shred proteins, carbohydrates, lipids, and DNA to pieces. And it involves reactive oxygen species, which are just toxic in general to any kind of compound. They're highly reactive, sort of small inorganic compounds that bind. They don't bind, but they actually chemically react with a large variety of things to cause damage. Okay, and we'll look at this more in detail in a minute. Secondly, when a PRR binds to a PAMP, it triggers the secretion of pro-inflammatory cytokines and chemotactic cytokines, or chemokines. For macrophages, there are 5 major inflammatory cytokines. They make tumor necrosis factor alpha, interleukin-1, interleukin-6, interleukin-12, and a chemokine that is referred to as CXCL8, or interleukin-8. And there are a lot of abbreviations in immunology, so try to get used to them early on. Tumor necrosis factor alpha is TNF or TNF alpha. And then all of the ILs, like IL-1, that is interleukin-1, interleukin-6, IL-6. Okay, I'm going to be referring to them mostly with abbreviations. These cytokines and chemokines activate other cells to promote a defensive state in the tissue and to promote inflammation. Right? Inflammation is sort of a defensive state. And lastly, binding of a PRR to a PAMP in some of the innate immune cells can trigger antigen presentation to CD4 positive T helper cells. Okay, so the macrophage is going to take up the pathogen into an endosome. It's going to shred the pathogen in that endosome, and once it has all those shredded little bite-sized pieces in the endosome, it's going to load those pieces onto MHC2 molecules and put the loaded MHC2 molecules onto the surface of the cell. There, T cells can swing by and survey the MHC2 molecules to see if they're foreign. And they are foreign if there's an infection, meaning that T cell is then going to respond by making cytokines that activate the macrophage. Okay, we'll come back to this all. Antigen presentation, loading of MHC2 is also going to involve the upregulation of a co-stimulatory molecule called B7 on the cell surface. And this helps T cells recognize that there's a problem. It's what we call a co-stimulatory molecule because T cells recognize that B7 is there. They have a receptor for it called CD28. And CD28 binds to B7. That tells the T cell that it can become activated, that there really is a problem here. Okay. Now, all of this, meaning the ability to express MHC2 and the ability to deliver co-stimulation to T cells, that defines macrophages as a professional antigen presenting cell, or PAPC. These are cells that can present antigen to T helper cells using MHC2. In addition to that, they also have to be able to provide a co-stimulatory second signal for T cell activation. Those things include B7, right? B7, cytokines, and other things. Okay. So, macrophages, they're present in the tissues or they are self-replicating. So you have populations of macrophages in your tissues that can divide and sort of provide their own growth stimuli. or macrophages can be produced from monocytes that have entered a tissue from the blood. Usually, this happens in large numbers when the tissue is inflamed, and so we need a bunch of macrophages to take care of the problem. A whole load of monocytes are going to leave the blood, enter that inflamed tissue, and differentiate into macrophages. During the infection, macrophages are going to stay in the tissues so that they can fight the infection. They're not going to move anywhere. Okay? This is in contrast to some other cells. After macrophages become activated by PAMPs and microbial products, or by cytokines or other danger signals like DAMPs, or damage-associated molecular pattern, they produce chemokines that attract T helper cells. They say, hey, come here and check this out. I have something to present to you. And then they present antigen in MHC2 to T helper cells. T helper cells respond by making cytokines that activate the macrophage to perform better killing. Okay. Macrophages make cytokines. They make antimicrobial products like reactive oxygen species and a number of enzymes that are deadly to bacteria, fungi, viruses, and parasites. And macrophages also partake in this thing called ADCC, which we'll talk about more later. So they look something like this. They're actually pretty modal cells. They chase things down, and they're really good at sort of seeing where the problem is, and they chase bacteria. They've been known to chase bacteria for meters and meters and meters. And if you stained a macrophage, it would look like this within the tissue. They're drawn sort of like this in Janeway. So yeah. Now here is a short list of some of the receptors that macrophages use to recognize microbes. We're going to be talking about these in more detail. But essentially you have cell surface receptors that recognize external pathogens. And then you also have intracellular receptors that recognize things that may be infecting the macrophage, or they may recognize things that the macrophage has taken up as being bad, as being a problem. Okay? We'll talk about each of these in detail later. And this is another view of some of the things that macrophages express on their surface. And when I say that, I mean transmembrane proteins that are being made by the macrophage, and they're kind of just sitting there as receptors, like in the membrane. Okay. You've got a lot of receptors that can cause the activation of the macrophage, multiple receptors for cytokines and other T cell molecules like CD40. You've got these different microbe recognition receptors like PRRs and such. And then you have receptors that can cause the uptake of pathogens right here. You have adhesion molecules that can allow the macrophage to stick to T cells and to stick to other cells. And then you've got peptide holders that cause activation of T cells. And the co-stimulator, of course, B7, that can deliver co-stimulatory signals to T cells. Okay. So here's a couple of ways that contents can be taken up by cells. Activated macrophages, dendritic cells, and monocytes, these are phagocytes, so they undergo phagocytosis. They have receptors that recognize that there is a pathogen, and then they sort of wrap pseudopods or pseudopodia around the cell and pull it into the cell in what we call a phagosome. Okay, so they're taking it up into a phagosome, and then that phagosome turns into an endosome, and it shreds its contents. Okay. Resting macrophages, dendritic cells, and monocytes, they haven't recognized anything bad. They're just taking stuff up and sort of cleaning out the tissues through penocytosis or macropenocytosis. This just means that they are taking up things that are innocuous, that aren't causing any kind of trouble. They're shredding them to pieces, yes, but they're not trying to present them to T cells. They're not trying to get any kind of T cell help because they haven't found anything scary. They're just clearing out the tissues of debris. Okay, that's part of their job. And then B cells, they have B cell receptors on their surface, and they can take things up through receptor-mediated endocytosis, where things are taken up into coated vesicle in the cell, and this is how B cells take things up. It's a little bit distinct from phagocytosis. Receptor-mediated endocytosis is how B cells take things up, how they take up pathogens. Okay. And after something gets taken up by a macrophage, it's trafficked through the cell. Okay. It's put into a phagosome here through phagocytosis, and then it sort of continues to move into the cell more and more. It moves toward the center of the cell in general. It becomes acidified and turns into an early endosome, which fuses with certain vesicles that are made by the Golgi, or trafficked by the Golgi and made in the ER. And those early endosomes contain things like those acid hydrolases that destroy pathogens and shred them to pieces. And they also contain MHC2. Okay. Later on, these early endosomes they turn into late endosomes, and that's really where most of the MHC2 is. That's being trafficked to late endosomes and then put out on the surface. But if it's not put out on the surface, it's instead trafficked to the lysosome, where you have really, really intense shredding enzymes and you have a lot of reactive oxygen species. The lysosome is kind of like a death pit for anything that is fused with it. Okay, so you've got a lot of trafficking of vesicles that occurs inside macrophages, and this is just kind of a view of that. Now, when you load MHC2, I have a figure on that actually. Maybe it's better to just wait until that. Here we go. So antigens can be taken up from the extracellular space into a vesicle in the cell, right? Endosome, phagosome, whatever you want to call it. You take up these particles, and those particles, they may have come from bacteria. They probably did come from bacteria, if we're talking about immunology, right? And in the early endosome, those particles that were taken up, they are at a neutral pH at first, okay? You have endosomal proteases present, but they're not turned on, okay? The proteases in the endosome have to be at an acidic pH to turn on. Later on, however, you have proton pumps that are just shooting hydrogen ions into the endosome, so it becomes very acidified. Its pH comes down as low as like 3 or 3.5, and that turns on those acid hydrolases. It turns on the proteases in the vesicle. Okay, and those proteases go to town on the thing that was taken up, and they shred it into little bite-sized pieces. And that pathogen is now a shadow of its former self. Okay, it's been broken up into many, many pieces. And it turns out that those pieces are a really good length for the MHC2 protein to bind to. So the MHC2 protein here, it is binding to the shredded remnants of the pathogen that was taken up, and it's going to be put out on the cell surface where a T cell receptor can come and bind to it. So another view of this is from a T cell perspective as opposed to a macrophage perspective. So a T cell wants to recognize this complex here. It wants to recognize a peptide that is inside an MHC2 molecule, or MHC1. Okay, so usually this peptide here is going to be buried inside the antigen as it normally exists. So that antigen has to be shredded into pieces, before it can be loaded onto MHC and have the T cell receptor recognize and bind to it. Okay. Then the T cell can provide that antigen presenting cell with cytokines, or if it is a cytotoxic T cell, and this is MHC1, it's going to say, hey, you need to die. You have a viral infection, I'm going to kill you. Okay, but if it's a T helper cell, it's going to be providing the macrophage with cytokines that activate the macrophage to become a better killer. Okay. And then we have neutrophils, which are kind of like macrophages, but actually a little bit different for macrophages. Neutrophils are also known as polymorphonuclear neutrophilic leukocytes, or PMNs, because they are neutral. They take up neutral dyes when you stain them, and because they have a polymorphonucleus, meaning their nucleus is segmented into multiple little pieces like this. And they have a highly variable appearance. Their nucleus has a highly variable appearance under the microscope. It's thought that the segmentation of the neutrophil nucleus helps with the squeezing of the cells between the vascular endothelial cells. The PMNs, the polymorphonuclear neutrophilic leukocytes, or the neutrophils. These are the first wave of cells that are recruited from the blood into a tissue where you have an infection. This is the very first cell type that gets recruited during an infection from the blood. So normally neutrophils are circulating in the blood, and that is where they are most concentrated. But then they're going to be called into an inflamed tissue so that they can help to fight the infection. Neutrophils are phagocytes that express pattern recognition receptors, but they cannot activate T helper cells. They don't express MHC2. They're not antigen presenting cells. Okay, they die pretty quickly. They're too short-lived to be a good antigen presenting cell. The recruitment of neutrophils from the blood is through that chemo IL-8 or CXCL8 that is made by macrophages that have been activated by bacteria or by a pathogen. Neutrophils can participate in ADCC just like macrophages. We'll talk more about that later. And they have a very short lifespan. They tend to circulate for 6 to 10 hours. Then they enter a tissue and they rapidly die. They usually live close to a day, but they have been known to live up to 6 days in the tissue. But regardless, they are going to die quickly. Okay, and they're going to leave a whole mess behind. So they're going to just leave their cellular debris and splatter it all over. And the macrophages have to, unfortunately, clean up all of that debris that the dead neutrophil left behind. Now, when neutrophils die, they undergo this process called netosis because they leave behind substance, a sort of thing called a neutrophil extracellular trap, or a net, N-E-T. Nets are basically just unchopped DNA. So normally when a cell undergoes apoptosis, it cleaves its DNA into a million little pieces, and that way it can nicely package its chromosome or its genetic material so that it can be disposed of by macrophages, right? However, when neutrophils die, they don't do that. They don't cleave their DNA. They leave it intact. And part of the reason for that is when they die, they can just release their DNA, and it sort of serves as like a trap that can capture bacteria or fungi or viruses or whatever and help prevent them from moving around as freely in the tissue and spreading rapidly. Okay? So when they die, they do not cleave their DNA, they just release it intact. Neutrophils are the main constituent of pus. If you've ever had a pustular infection, you know that pus is pretty white or green, depending on how gross it is. But most of what you'll find if you look at pus under the microscope is you will find dead neutrophils. You will find dead neutrophils. And you have so many neutrophils recruited into an infection that they accumulate as pus like that. Neutrophils are a member of this group of cells called the granulocytes because they have densely staining intracellular granules like this that you can actually physically see each of the little granules under the microscope if you have a good eye. Okay, so they are granulocytes, and there are other cells that are considered granulocytes, but this makes them distinct from the macrophages, monocytes, and dendritic cells because those cells are not granulocytes. Okay? And neutrophils have segmented nuclei. That's really what best defines them. It's a very classic appearance. So here's what they look like in the blood. They have those segmented nuclei. And here is what the nets look like, the neutrophil extracellular traps. All of that blue stuff is uncleaved DNA that has been released from dead neutrophils. The red stuff is what can help tie together all of the released DNA. And that red stuff is actually neutrophil elastase, which is sort of sticking to itself and is also sticking to the DNA. So this is what nets look like, NETs. This can help capture bacteria kind of like a spider web and prevent it from moving very far. Okay, so NETs just trap bacteria. And that is where I'll leave off for today. So I'm going to stop my recording.